Hello everyone, my name is Shiyu Xiao. This video will talk about three Lac operators and their effect in repression, specifically regarding Figure 7.10 and 7.11 from Molecular Biology 5th edition book by Robert F. Weaver. This video is made for MCDB 427 Molecular Biology at University of Michigan. So first of all, what is operon? In genetics, an operon is a functioning unit of DNA which containing a cluster of genes under the control of a single promoter. Basically, operons are common in bacteria, but they are rare in eukaryotes such as humans. There are several typical operons like Lac Operon, Ira Operon, Trip Operon, and Gal Operon. In this video, we will focus on the Lac Operon, which is known as the first operon to be discovered. Here's the introduction of Lac Operon. This is the regulatory element for the Lac Operon. There are two kinds of components. There's regulatory site, which contains regulatory gene LacI, cap binding site, the promoter LacP, and the operator LacO. Also, structural genes lacZ, lacY, and lacA, which encodes for beta-galactosidase, galactoside permease, and galactoside transacetorace. Next, we will briefly go through the negative control of the Lac Operon. In 1940, Francois Jacob and Jacques Monard of the Peugeot Institute showed the on-off nature of lactose utilization. So first of all, this is the condition which lactose is absent. If a culture of E. coli is growing in a medium lacking lactose, the intracellular concentrations of beta-galactosidase and permease are extremely low. In this condition, no Lac gene is expressed. LacI gene has its own promoter, and its expression can produce LacI repressor. Then, the LacI repressor monomer can form the tetrameric LacI repressor, and this tetramer binds to the LacO site. This binding blocks the RNA polymerase moving on DNA template, and no lacZ, lacY, and lacA are expressed. Second, this is the condition which lactose is present. If lactose is added to a culture of E. coli in a lactose-free medium which also lacks glucose, both beta-galactosidase and permease are synthesized simultaneously. In this condition, lacZ, lacY, and lacA genes are expressed. Beta-galactosidase is weakly expressed at a basal level. When lactose is present, it is converted to allolactose or galactose that bind to the repressor. Here, I want to emphasize that the LacI is allosteric protein, therefore the binding of the Lac Operon inducer, here referred to allolactose, to the LacI repressor changes the shape of LacI, and it can no longer bind to the operator. In the lack of repression, the Lac gene can be expressed. One more note is that galactose, allolactose, and IPTG are referred to as inducer. So, be more specifically from this biochemistry point of view, we can see that when beta-galactosidase cleaves lactose to galactose and glucose, it also rearranges a small percent of lactose to allolactose. So basically, beta-galactosidase will change beta-1,4 linkage in lactose to a beta-1,6 linkage in allolactose. Then this allolactose will bind to the repressor tetramer, and as we can see in the previous slide, that the inducer removes the repressor from the operator, allowing RNA polymerase to transcribe the structural genes. So as emphasized here, the control of the Lac Operon, the way I just described, is incomplete, as it only talked about one LacI binding site, one operator. In fact, there are three different operators involved in controlling the Lac Operon. There are one major operator near the transcriptional start site and two auxiliary operators, one upstream and one downstream. This is a complicating factor in repression of the Lac Operon, and this is also the part that many scientists are interested in. So now, after we finish going through all of these concepts, we will move on to the key part of today. Actually, the Lac Operon contains 3 binding sites for LacI repressor. In figure 7.10, it shows the map of the Lac control region and the sequence of the 3 operators. The numbers given here are relative to the transcriptional start site. The classical operators LacO1 is centered at position +11. The downstream auxiliary operator LacO2 is centered at position +412. Here we note that operators can be at downstream as well. The upstream auxiliary operator LacO3 is centered at position -82. The cap and RNA polymerase binding site are in yellow and blue respectively. So this is the sequences of the 3 operators. We can see the central G of each labeled in boldface. Those sites at which the auxiliary operator sequences differ from the major operator are labeled in lowercase in the O2 and O3 sequences. Now, you may ask which operators are important. Are these auxiliary operators necessary? Mueller-Hill and others in 1990 have discovered that auxiliary operators are not just trivial copies of the major operator. Instead, they play a significant role in repression. In order to determine whether all 3 of these operators were important for the control of the operon, the investigators mutated one or more of the operators and assayed for lacZ expression. Mueller-Hill and Corrigus constructed 7 different operator mutants. In this figure, the crossed bar or the arrow indicated part are those destroyed operators or inactivated operators by doing mutation. Here's the respective mutations they made in the operator sequences. The wild-type sequences of O1, O2, and O3 come from the figure 7.10 which we see before, and the red circled nucleotides are the ones that did mutation. The red highlighted part in the mutant sequences are the altered nucleotides. After they made constructs with one or more mutated operator sequences, the second step is to clone these mutants and the wild-type DNA into lambda-phage. Lambda-phage packaging is very efficient. It can be 1,000 times more than transformation, and also lambda-phage can commonly package 12 to 20 kilobase of foreign DNA. Then, using this new cloned lambda-phage to infect E. coli and allow these DNAs to lysogenase E. coli cells. As a note, lysogenic here means integrates into chromosome. After that, they assayed beta-galactosidase activities in the presence and absence of the inducer 1 millimolar of IPTG, and then calculate repression, which is ratio of activity in the presence and absence of the inducer IPTG. So as I emphasized, here's the fourth repression, and they got by beta-galactosidase activity in the presence of inducer, which means plus IPTG condition, and divided by beta-galactosidase activity in the absence of inducer, which means minus IPTG condition. So now we can see what results and conclusions they got from this experiment. These auxiliary operators play a major role in the repression of the Lac Operon. First of all, when none of the LacO exists, there's no repression, which for the repression indicated by 1.0. Now we can see when all of the 3 sites are present, LacI repressor can repress initiation of transcription approximately 1,300-fold. We can see that missing LacO2, the repression is 440-fold, and missing LacO3, the repression is 700-fold. So, not changing LacO1, if either LacO2 or LacO3 is missing, the repression is at about 500-fold, which indicates that you don't need all of the 3 operators to get good repression, just LacO1 and another operator. Then, if both LacO2 or LacO3 are missing, repression is only 18-fold. You get some repression from Lac Operon alone, but nothing like when another is also present. We can conclude from here that removal of either of the auxiliary operators decreased repression only slightly, but removal of both auxiliary operators decreased repression about 50-fold. Then let's see the condition which under removal of Lac O1 and change or not change Lac O2 and Lac O3. We can see that after remove Lac O1, they only have 1.9-fold repression even both auxiliary operators exist. The repression is almost equal to no repression under removal of all of the three Lac operators. Then we see after remove Lac O1, only keep one auxiliary operator, either Lac O2 or Lac O3. There's no repression. So we can conclude from here that Lac O1 is crucial among three Lac operators. Lac O2 and Lac O3 alone is not working. So now here's the overall conclusion. First, Lac O1 as a classical operator, it is essential for the repression. Second, Lac O2 and Lac O3 as auxiliary operators, they help Lac O1 to get good repression. Third, missing either Lac O2 or Lac O3 is fine, but removal of both auxiliary operators decreases repression about 50-fold. Furthermore, we can talk a little bit more based on that. So to recall the memory from previous slide, the tetramer of LacI I showed early in the video, actually it not binds as tetramer like this. There have two binding sites, and each binding site binds to LacI. Basically, when the LacI tetramer bind to Lac O1 and Lac O3, two of the monomers bind to Lac O1 and two of the monomers bind to Lac O3. This causes looping of the intervening DNA that further reduces the binding of RNA polymerase to the promoter. When the LacI tetramer bind to Lac O1 and Lac O2, two of the monomers bind to Lac O1 and two of the monomers bind to Lac O2. This causes looping of the intervening DNA that directly interferes with mRNA initiation and elongation. Also, there are other factors that can influence the regulation of Lac Operons, such as CAP is a positive regulator of the Lac Operon. However, they will be discussed in the next section of this chapter, so I will not talk more about that. And for this video, we only focused on three Lac operators and their effects. We can also see the structural basis for the cooperativity among operators which provided by Michel Ruiz and co-workers in 1996. The structure present in pink, green, yellow, and red are the four repressor monomers, and the structure present in blue are the DNA fragments. We can see that two repressor dimers interact with each other at bottom to form tetramers. Each of the dimers containing two DNA binding domains that can be seen interacting with the DNA major groove at top. The structure shows clearly that the two dimers can bind independently to separate Lac operators. I hope this video helps you better understand three Lac operators and their effects in repression. Thank you for watching, and as always, go blue!